going to talk about today is a project we've been working on for quite a few years now regarding the development of the new immunotherapy for cancer, where we use antibodies to target macrophages. So I will give you a little bit of background first to this project. So we've known, immunologists have known that the immune system can recognize cancer for over 100 years. But it's only the last 10-15 years that this actually has come to clinical practice. And the reason for this is that it's taken quite some time to figure out exactly how the immune system works and which points we can actually push on the immune system to make it do things to help us in the clinic. And what really changed things were when we figured out that T cells need co-stimulatory receptors. So there's two types of co-stimulatory receptors. There's inhibitory and there's activating receptors, where classical treatment could increase survival, but now together with these so-called checkpoint inhibitors, PD-1 and PD-L1, we can actually cure part of the patient group. And also in combination, you can cure even more people. And some cancers are then more immunogenic than others. And you see that melanoma is on top there, which acquire most mutations. So this is more easily recognized by cells and a lot of different immune cells that are regulated and also are used by the tumor to actually block growth from embryogenic progenitors. And the cell type called for M1, wound healing for M2, and regulatory for capacity of macrophages. And on the other hand, you have the immune blockage of macrophages. And you can see to the right there, and the immune system have various ways to recognize these immune systems. But equally important is dangers. So where you have stress tissue or damaged tissue, this will also, for instance. And we have a number of different epitopes here that you can also find on microbes. And the immune system will recognize these by natural antibodies, but also through specialized receptors. And one group that I'm going to talk about today is called scavenger receptors. So, and in particular, I'm going to talk about the scavenger receptor called Marco. So this is Marco. It's here in the center, the very tall receptor. It's a trimeric receptor. It's expressed on macrophages. It has a long collagen domain. And then in breast cancer, they looked at different stromal genetic signatures and see if any of these were connected to good or bad prognosis. And very high up on this list, we actually found Marco. So this receptor that we were working on in the complete models. And then you have a colon cancer model and a melanoma model. And in all these cases, we found that Marco is indeed expressed in the tumors. So they are thought to be a population that are more sort of perivascular type macrophage, immunosuppressive macrophage. This was also true when we looked into the tumor itself. So here we have B16 melanoma tumor. So did this have any effect then on tumor growth? And it turned out it did. So here is now the breast cancer. This is the 41 model. Just one injection, more of these M1 type macrophages and a little bit less of these anti-inflammatory M2. So there was a shift in the macrophage. By targeting these macrophages, we would change the vessels within the tumor. So we collaborated with a lab where we did actually staining of the whole tumor. So we stained. So we don't think that we actually affect this capacity of macrophages. The second thing we did fairly early on is that we tested whether this treatment would actually work together with these T-cell targeted therapies. So here we have given either anti-CTLA-4 antibody alone or together with anti-Marco, and you can see that it actually enhances the response. This is also true if we do here in the EO771. We tried anti-PD-1 or anti-PD-L1 together with our Marco, and you can see that Marco by itself is as good as PD-1 or PD-L1, but in combination we even get a better. Second question then is, what is actually happening here? Are we enhanced? And when we did this, you can see that here actually the treatment still works. So this was a big surprise to us. So CD8 T cells, including also unconventional T cells. So then our sort of thoughts turned towards NK cells. Because we did see with anti-Marco treatment that we got an influx